In this short video, we're going to go over how to interpret the results of hits to the conserved domain database. So here we are on a BLASTp output page that we're used to seeing. And not all the time, but in this particular case, we have a hit to conserved domain database. How do I know that? I see this little graphic here above my BLAST hit visual that shows and it says putative conserved domains have been detected. Click on the image. So I can go here and click, and in doing so, I pull up a little more detail about the hits to what's known as the conserved domain database at NCBI. So this makes use of multiple different types of models of different protein domains, some of which we'll cover here and some of which we'll hold off on until we get to another module. So what we see is a visual here of different pieces of information, some of which cover large stretches of protein. You can see I can mouse over it and I pick up different information, and some of which are very specific to certain residues, like here's an ATP binding site, and I can put my cursor over that and learn a little bit more about it. So the two types of domains that we're going to work with here, and this is within the module on sequence similarity, are hits to COGs and hits to conserved domains. All right. So here is a COG hit, COG 1204. In fact, it's the best hit here of all the ones shown. This particular COG is for something called replicative superfamily 2 helicases. And if we hit this plus button, we can open up a little bit more information. We can see how well our sequence matches the COG model. Now, COG stands for a cluster of orthologous genes, and it's based off a minimum of 3 proteins from 3 different organisms who are each other's reciprocal best blast hits in 2 different genomes as you compare them to each other. And in doing so, they represent some conserved protein found across at least 3 different genomes. And this was first done back when there was only like 10 genomes out there. And this was a way of trying to identify key proteins that maybe was shared by much of life, if not all of life. That still is true, although because we've sequenced so many genomes, we might find some COGs that are unique to certain smaller groups. So in this case, we have a hit to a COG that tells us something about our protein being a helicase, and that in fact agrees with our BLAST results. So I'm going to go ahead and close that one back up. I'm going to look down here and I see that I have another hit, still quite good, to a conserved domain that says DAD like helicases, superfamily. And I can open that up. It tells me a little bit more about them. These are a diverse family of proteins involved in ATP-dependent RNA or DNA unwinding. And again, I can see how well it matches, in this case, along the full length of the conserved domain. So these are some additional pieces of information that you can use to help back up what you're seeing from your BLAST analysis. And with that, that ends this short video.